After watching this video, you should be able to describe the key components of steroidogenesis, list the major steroid hormone classes and key examples, the locations of where steroid hormone synthesis occurs, and the detailed steps of making steroid hormones from cholesterol. Now, let's start with the steroid hormone classes, and we can see we have 1, 2, 3, 4, 5 major classes of steroid hormones. Now, each of these hormone classes, the way to think about it is that they're defined by the receptor that they activate, which then dictates the physiological effects of these hormone classes. So we start with glucocorticoids. The prototypical example is cortisol. It binds to and activates glucocorticoid receptors, abbreviated here GR. Now, mineralocorticoids, the main one is aldosterone, and the receptor is the mineralocorticoid receptor. But there are other mineralocorticoids, other hormones that can activate the mineralocorticoid receptor. Usually this is under pathophysiological conditions, and one example here is 11-deoxycorticosterone, which is a precursor in some of the steroid pathways that we're going to look at. And when this hormone builds up, and typically this is because there's an enzyme defect that we'll talk about later, or sometimes rarely there's hormone secreting tumors that make lots of 11-DOC, we can end up with a patient that looks like they have high aldosterone from mineralocorticoid effects, but in fact their aldosterone is not the source of the problem. It's actually something else, in this case 11-DOC. Now, it is worth mentioning also that cortisol can activate the mineralocorticoid receptor, and it really doesn't have effects at the mineralocorticoid receptor normally, because in the kidney, cortisol is locally inactivated to cortisone. So we also could put cortisol here as another hormone that could activate the mineralocorticoid receptor, but we don't typically see that under physiological conditions. Okay, now progesterones is another class, and there's only really one important progesterone, part of the progesterone class, and that's just progesterone. And it activates progesterone receptors. I just summarize them all here as just PR. There's different subtypes. And the progesterone receptor is going to have some important effects in as a sex steroid. Now, the rest of the sex steroids here, we can see that there's multiple examples. We have androgens, and we see here there's DHEA, Androstenedione, testosterone, and dihydrotestosterone, and all of these hormones are in the androgen class because they all are capable of stimulating the androgen receptor. Now, they have different activities and androgenic effects. DHEA and androstenedione are considered weak androgens, and testosterone and dihydrotestosterone are the major androgens that cause the androgenic effects by stimulation of the androgen receptor. Okay, now the final class here are estrogens, and there's multiple ones. In fact, there's even an additional one called S-tetrol, which I don't have listed here. But these are the three main ones that people think about with this class. And again, these are considered estrogens because they activate estrogen receptors. Again, there's multiple subtypes of estrogen receptors. We're just summarizing them all here with the abbreviation ER. Okay, so it's important to know these class of hormones and these examples and the names of the receptors. Now, for the locations of the steroid hormone synthesis, the adrenal cortex is very important. The adrenal cortex, remember, is the outer layer of the adrenal gland that surrounds the adrenal medulla, and there's actually 3 zones that have different hormones that are secreted that we'll talk about in a little bit. The gonads, the ovaries, and the testes, they have specific cell types that express different enzymes and make different hormones that we'll talk about a little bit. And the placenta is a very important source of steroid hormones during pregnancy. Now, in order to make steroid hormones, we need to derive them from cholesterol, and this is going to require a little bit of chemistry just to show why the names of the enzymes have have their names that they do. And I think it's helpful when you're learning the pathways. So let's take a look next now at cholesterol. Okay? So cholesterol is the precursor of all steroid hormones. And we can see here that it has these 4 ring structures that are fused, labeled A, B, C, and D. Okay? And there is a hydrocarbon chain coming off over here. And we can see that the total number of carbons is 27. Okay? Now, where does this cholesterol come from? Well, there's lots of different ways that the steroidogenic cells can get cholesterol. A major one is uptake from LDL cholesterol via the LDL receptor. Recall that the LDL receptor engages the LDL that contains the cholesterol esters and undergoes receptor-mediated endocytosis, taking in the cholesterol into the cell. Okay, and then now the cell can use the cholesterol for steroid hormone synthesis. There are other possibilities. Some of these cells have the capability to make cholesterol de novo from acetyl-CoA. And another mechanism would be that there are pools of cholesterol esters that are formed by ACAT, acyltransferase, um, that takes acyl groups and esterifies the cholesterol, and that serves as a active pool of cholesterol that can be tapped into. Okay, so regardless of how you get the cholesterol, we end up with it here, and we can see that this first step, which I've labeled P450 side chain cleavage, is the rate limiting step. And you can see that the molecule that we make here is pregnenolone, and the only difference between the two molecules, cholesterol and pregnenolone, is we can see that we've shortened the molecule down to 21 carbons. Okay, so we've cleaved off the side chain. Okay, and now we're left with this molecule over here. Now, before we go and look at the details, I want to highlight the numbering system on this cholesterol molecule, which then transfers over to all the other molecules that are made downstream. Notice that there is a hydroxyl group at carbon number 3, and this carbon number 3, there's going to be some action going on, as we're going to see some of the hormones that are formed are going to have some changes occurring at carbon 3. We're also going to have some changes that occur at the 5 carbon and this double bond here. We're going to see some changes there. We're also going to see some other carbons that are going to become important. So, for example, the 11 carbon, okay, the 15 and 16 is especially important. It can be modified in when we talk about placental hormones and what happens in the fetus. The 17 and 18 carbons are also going to be a site of action, okay, and the 21 carbon. Okay, so those are all places that you should keep your eye open for as we're going to start to talk about some of these enzymes. They're going to be using these numbers, and this is where on the molecule that we're going to be having modifications. So when we're talking about steroid hormones, the other thing to think about is we're mostly just talking about hydroxylases, which are going to add OH groups, and dehydrogenases, okay, that are going to remove hydrogen. So with the exception of a couple of other enzymes, those are the major enzymes that we're going to see, and it's going to be a theme as we make these hormones. Okay, so now let's take a look now at the chemistry of these molecules. Now I want to direct our attention here to cortisol. All right, let's take a look at what's different with cortisol compared to pregnenolone. Look here. I have on this 3 carbon, instead of having an OH group, I now have a double bond O. And in this A ring, I now have moved over this double bond that was in the B ring, and now it's shifted over. Okay, so that's a change. That's something that's different. I also can see that I have a 17 hydroxyl. Okay, that's not something that we saw in pregnenolone. I also see a 21 hydroxyl group, okay, and I see an 11 hydroxyl group. So remember we were talking about dehydrogenases and hydroxylases. So if I was going to guess, I'm going to be needing a 3-dehydrogenase, a 17 hydroxylase, a 21 hydroxylase, and an 11 hydroxylase. Okay, so just based on how this looks, I can figure out what enzymes I'm going to need. Okay, now if I go and look at aldosterone, you can see that it's really not a whole lot different. We can see that the 3 carbon also has the double bond. The double bond O. We can see that the double bond shifted over like it did for cortisol from the B ring to the A ring. We see that there's a 21 hydroxyl. Okay, I see that there's 11 hydroxyl, but look, there's a difference. There is no 17 hydroxyl group, but the 18th carbon has something different going on there. There's actually an oxygen here that is bonded to carbon. And so that is different than cortisol. Cortisol didn't have anything with the 18 carbon that's changed. It's just a methyl group there. So if I was going to guess for aldosterone, I would guess there would be a 3-dehydrogenase, a 21 hydroxylase, some sort of 11 hydroxylation, and something with the 18th carbon that I would expect. Okay, so just based on how these look, we can start to predict these things. Now, if I go look at a progesterone, all right, which is not that different than pregnenolone, I don't see any hydroxyl groups at all with progesterone. The only thing I see is this OH at 3 that was converted to a double bond O, and the movement of this double bond from the B ring to the A ring. So I would expect that I would have again a 3-dehydrogenase, and that would really be the only thing. Okay. Now let's take a look at testosterone and dihydrotestosterone, the androgen class. Now, if I see these guys here, okay, again I see the double bond O in the 3 position. I see the double bond that was shifted over. I don't see any other hydroxylations. There's nothing going on at the 18. There's nothing going on at the 11. But what I do notice is that at the 17 carbon, okay, there is no hydroxyl there anymore. In fact, what I see is a reduction in the number of carbons in this molecule. See over here, for these molecules, progesterone, cortisol, and aldosterone, these are 21 carbons in length. In fact, they're commonly referred to as C21 steroid hormones. C21 indicating they have 21 carbons. The testosterone here and the dihydrotestosterone, they're only 19 carbons in length. So they're commonly referred to as C19 steroids. Okay? And so something happened. Something there was a lyase to the 17 to these carbons over here, clipping them off, leaving the 17th carbon alone here, minus these 2 carbons. Okay? Now, the dihydrotestosterone, the only difference I see here between testosterone is that I've eliminated this double bond between these carbons here in the A ring, and now there's just a hydrogen there. So this is dihydrotestosterone. Something happened at that 5 carbon. There was a reductase probably that acted there. Okay, and then finally for estradiol as our estrogen example, you can see that it looks very similar to these androgens, except that in this A ring, there's an aromatic ring. There's no double bond anymore, and there's a 3-hydroxyl group. All right, and I lost a carbon. Okay, so estrogens are commonly referred to as C18 carbons for that reason. See, we lost that carbon right there. Okay, that we had in the androgens, and frankly the other steroid molecules. So there has to be an aromatization of this ring, and we're going to see that that's going to be coming directly from testosterone if we're talking about estradiol. Before we go on to the specific steps, I just want to point one other thing about estradiol. The estrogens, the naming convention of them is based off of the hydroxyl groups. So you see how there's 1 hydroxyl, 2 hydroxyls. that's a diol. Okay? If I just have one hydroxyl group, for example, if there was a ketone here in this D ring, and it was just one OH in the A ring, that would be called estrone. Okay? And actually, if there's another hydroxylation, commonly the one that we'd be thinking about would be at the 16th carbon. There'd be 1, 2, 3. That's called estradiol, which is a very important estrogen of pregnancy. Okay, so it may not be something that you'll readily be thinking about in terms of the chemical structures when you're thinking about medical examples of these pathways, but I think it's good to at least look at these one time to see where these enzyme names come from and how these molecules are different. Okay, so now let's take a look at the pathway, and what we're going to do is we're going to get rid of the chemical structures and we're just going to use the names. Okay, so the first place that we're going to look at is the adrenal cortex as an example, and we can see there's a lot going on here. There's a lot of enzymes and there's a lot of individual steps and precursors. So let's just start with making aldosterone. Now, aldosterone is made in the zona glomerulosa, which is the outer layer of the adrenal cortex. And in order to make aldosterone, we're going to take cholesterol and we're just going to go straight down, right there to aldosterone. So we're going to need our side chain cleavage to make pregnenolone. And then I have this enzyme 3-beta hydroxysteroid dehydrogenase. Remember, we were talking about in that A ring, right? We were talking about getting that, going back for a second, we were talking about getting that OH that was originally in cholesterol and pregnenolone and making a double bond. Okay, so that's what that enzyme is there for. Now, why is there ISOM in this name? Well, this is a bifunctional enzyme. Not only is it going to do a dehydrogenase, but it's also going to do a isomerase, and that's why that double bond moves from the B ring to the A ring. Okay, so this is an enzyme that's going to be doing those two functions, and when we do that, we make progesterone. Now, remember when we were looking at progesterone, that was the only difference that it had with pregnenolone. It only had that OH to double bond O and that double bond from the B ring to the A ring. So there's our progesterone. Now what we're going to do is we're going to hydroxylate it to 21st carbon. And when we do that, we make a molecule called 11-deoxycorticosterone, which we said has mineralocorticoid activity if it accumulates under pathophysiological states. Normally, just flows down and makes, and in the case of the zona glomerulosa, it just goes to corticosterone. And the enzyme that does this first conversion to hydroxylate the 11 carbon, okay, is called P450 aldosterone synthase, which is abbreviated here P450 aldo. Okay, now P450 aldo is interesting because it has three functions actually. So it does an 11 hydroxylation, and then what it does is it does an 18 hydroxylation to set up an oxidation reaction to make that double bond O in the 18th carbon. Okay, so see what we needed to make aldosterone. We needed a side chain cleavage, of course, because pregnenolone is the precursor parent compound to all the steroid hormones. We needed our 3-beta HSD isomerase. We needed the 21 hydroxylase and the three functions of aldosterone synthase: 11 hydroxylase, 18 hydroxylase, 18 oxidase. Now, for the zona glomerulosa, that zone lacks the enzymes to go to the right here. So everything flows down the aldosterone in the zona glomerulosa, and it does not have P450C17 functionality there. Okay, so that's a pretty straightforward layer, and we'll talk more about that in another video in depth about the adrenal cortex. Now, let's make cortisol next. Now, cortisol, the thought is, the easy way to think about it is this is mainly made in the zona fasciculata. Some texts and some individuals just like to think of cortisol just being made by the zona fasciculata and reticularis. But for the purpose of this video, let's just say this is happening in the zona fasciculata. Okay, the next layer in the adrenal cortex. Now, how do we make cortisol? Well, remember when we looked at the molecule, right? We needed, of course, to have our side chain cleavage to make our pregnenolone. right? And then what we needed was we needed to have a 17-hydroxylation. So let's go this way. Go to pregnenolone to 17-hydroxypregnenolone. And that's done by this P450C17 enzyme, which the first function is a 17-hydroxylase. Okay, so there's our 17-OH pregnenolone. Now I need to convert it to 17-hydroxyprogesterone with the 3-beta HSD isomerase enzyme. Now I have 17-OH progesterone. And then from there, I hydroxylate it to 21st carbon, making 11-deoxycortisol. And then I put the final hydroxylation on the 11th carbon via another enzyme called P450C11 11-hydroxylase. Now remember that 11-hydroxylation in the zona glomerulosa, that was done by the aldosterone synthase enzyme. This is the same effect, but it's a different enzyme. Okay, different enzyme having the same effect. And that final hydroxylation gives us our molecule cortisol. Okay, so remember we were looking at these molecules. Remember we were looking at cortisol. Remember there was our 3-beta HSD isomerase right there. Okay, having its effect there. There's my 17-hydroxylation. There's my 21-hydroxylation. And then there's my 11-hydroxylation. And there's cortisol, our prototypic glucocorticoid. Okay, now if you wanted to make cortisol from pregnenolone to progesterone and then go over to make 17-hydroxyprogesterone and then go down, you certainly could do that. That doesn't really make any difference. We're still going to be always flowing down to cortisol. Okay, now also what's made in zones other than the zona glomerulosa is 11-DOC. Remember, it was made to corticosterone in the process of making aldosterone in the zona glomerulosa. I also can make some corticosterone in the zona fasciculata and probably the reticularis as well. Okay, but the reason why we're focusing on cortisol is that's a biologically active glucocorticoid that is clinically relevant. Okay, now let's make androgens, these guys over here on the far right. And if we wanted to do that, you can see that what makes these molecules, and I've only listed here DHEA and androstenedione because those are the main adrenal cortex androgens. Okay, the reason why they're special and different than all the other ones here is that, remember when we were looking at the chemical structures, that they were missing 2 carbons, right? You see right over here, I lost these carbons, okay? I lost 2 of them. So if we go back over here, I'm going to need a lyase enzyme. And so what does that actually is the P450C17 enzyme. It has a second enzyme activity that's required to make androgens. And so if I want to make DHEA, it's pretty simple. Okay, I do my side chain cleavage. I have pregnenolone. I make 17-hydroxypregnenolone. And then I make DHEA. Okay, so I just need really 2 enzymes, side chain cleavage and P450C17 with its 2 functions. If I want to make andro, okay, androstenedione, I could take DHEA, all right, and then I can have it undergo the 3-beta HSD isomerase reaction and make andro that way, or I can take 17-hydroxyprogesterone and make androstenedione from there and have the 1720 lyase function. Okay? So if you want to think about the zona reticularis making these hormones, you can appreciate then that these pathways would be going to the right. Okay? They'd be going to the right and you'd be making these. And actually the principal adrenal cortex androgen quantitatively is DHEA, but qualitatively androstenedione is more active than DHA and is one step closer to making testosterone and DHT. Okay, so that's how you would make these hormones. And the reason why these enzyme names and even some of these precursors are really important is because there's very important clinical relevance. So let me give you a few examples to think about. So there's a host of disorders called congenital adrenal hyperplasias, which we'll have another video on. And all of those disorders under that umbrella term, congenital adrenal hyperplasia, have one mechanism in common. There's an enzyme defect, a congenital defect, in the steps to make cortisol. And if you look at cortisol, remember, it was a complicated synthesis. There was multiple steps, okay? I had an 11 hydroxylase, I had a 21 hydroxylase, I needed a 17 hydroxylase. And of course, I needed my 3-beta HSD isomerase and side chain cleavage. Well, the most common cause of CAH is 21 hydroxylase deficiency. Okay, and you also can get it, it's rare, but you also can have 11 hydroxylase deficiencies and 17 hydroxylase deficiencies. In any of these cases, cortisol synthesis is inhibited, right? Because if you have any of these enzymes defective, cortisol levels go down. Now, you also, of course, could have a 3-beta HSD isomerase and side chain cleavage defects. Those also would affect cortisol, but the major ones that people should know are these hydroxylase defects. Now, what happens when cortisol goes down is we'll discuss this in another video. Because of lack of negative feedback from cortisol on the glucocorticoid receptors, adrenal corticotropin hormone goes up from the anterior pituitary, and that stimulates the adrenal cortex to make more hormones. Now, what happens is you're going to start to build up precursors, and they're only going to go in the direction that the pathways that are available that they can go in. So if I had, let's say, for example, a 21 hydroxylase deficiency, then the only place for me to go is to make androgens. And you can imagine that if it's a fetus that's XX, female fetus, it's going to be exposed to a lot of extra androgen, and that's going to masculinize and potentially virilize the fetus. In addition to that, not having a 21 hydroxylase enzyme is going to cause problems just from lack of cortisol. And if the defect is severe enough, you can see that the 21 hydroxylase enzyme is also responsible, right? It's also found in the zona glomerulosa. So you can actually have to start to have aldosterone problems as well. So there, again, there's going to be another video where we'll go into these in more detail, but you should appreciate that if an enzyme is knocked out, what hormone are you not going to make, and then what molecules or hormones are going to build up, okay? Another example is we were talking about 11-DOC, right? Well, what would make that increase? Remember, we said we don't typically have that doing anything physiologically in most cases, but if I had 11 hydroxylase defect that caused cortisol to be synthesis to be inhibited, I also would end up with an accumulation of 11-DOC. Okay, not in the zona glomerulosa, but in the zona fasciculata or maybe even the reticularis. Okay, so and then that would again, cause effects in the mineralocorticoid receptor. All right. Now, there's also individual precursors that don't really have biological effects, but are very important hormones that are measured to diagnose disorders. Like, for example, that 21 hydroxylase deficiency that I was telling you about, it's the most common site of CAH. If you have that enzyme inhibited, the immediate precursor, 17-hydroxyprogesterone, is elevated, and that's one of the tests that you can do to see if someone has the 21 hydroxylase defect, because you would imagine that the immediate precursor that's acted upon by the enzyme would build up. Okay, so a lot of these steps are really important, especially in disease, and it's a really good idea to be thinking about them and using this pathway. All right, now the final part of this video is we're going to talk a little bit about the gonads. Okay, the ovaries and the testes. Now, if you see in here this picture, what I have removed is aldosterone and the cortisol pathways, and the reason why is because the gonads, they don't make those hormones, all right? So what they do is they make sex steroids, right? The androgens here in magenta, okay, and the estrogens here in pink. Now, we're very familiar with these enzyme pathways, so let's start with our androgens that we were discussing. So here's DHEA and androstenedione, okay, that would be precursors then to the more potent sex steroids, more potent androgens, testosterone and DHT. Now what's happening here with androstenedione is I'm using a 17-beta HSD, hydroxysteroid dehydrogenase, to make an OH group at that 17 carbon. So if we go back to our structures, okay, notice that there's an OH group right here. Okay, now that OH group is not there in androstenedione or DHEA. It's a ketone, right? So we need to make that an OH in this case, and that's a 17-beta hydroxysteroid dehydrogenase. Okay. Now, once we have our testosterone here, the testosterone, I'm going to make that. If we go back here again, the difference between testosterone and DHT is in that A ring. I'm going to get rid of that double bond, okay, and then there's my hydrogen there that's added, and that's a 5-alpha reductase step. Okay, that's this enzyme here, and I make this hormone DHT. Okay, dihydrotestosterone. Now I also can make estrogens, and estrogens, you need to remember, are made from androgens. And you can see here, if I have androstenedione, when I have an aromatization of androstenedione, I only end up with one OH, so it's estrone, which is a weaker androgen. Weaker estrogen. And then testosterone, if it's aromatized, I have my two OHs, and I have estradiol. Okay, so estrogens are made from androgens. Okay, and in this case, you should know estradiol from testosterone, estrone from androstenedione. Now, what's not shown here is that DHEA can also play an important role in providing androgen precursor to the production of estrogens. Now, particularly that's important in the fetal adrenal gland. Okay, so in the fetal adrenal gland, has a special zone that doesn't have the 3-beta HSD isomerase, and the fetal adrenal is making lots of DHEA. And that DHEA goes and gets hydroxylated at the 16th carbon and goes to the placenta, where it's aromatized, and you make something called estradiol, which is a very unique estrogen of pregnancy. So even though this is in the Gonads section, you think reproductive sex steroids, I don't have the placental fetal unit. We'll show that in another video. But I did want to point out that DHEA can be further hydroxylated, and then that could provide a precursor for placental aromatization. Okay, so going back to where we started, we discussed steroid hormone classes, glucocorticoids, prototypically cortisol, mineralocorticoids, aldosterone, and you'll see in disease states 11-DOC, particularly enzyme defects and congenital adrenal hyperplasia. Progesterone, acting at the progesterone receptor, all are various androgens, and you should understand that DHEA is the precursor to androstenedione, which is the precursor to testosterone, which is the precursor to DHT, and really the testosterone and DHT are the active androgen, the more active androgen at the androgen receptor. We also have the estrogens, which come from androgen. They're aromatized. Remember that A ring is aromatized. I lose that methyl group, and I have 18 carbon molecules. These are all acting at the estrogen receptors. Estradiol is probably the most biologically active and relevant estrogen. Estradiol is important in pregnancy. We'll discuss that when we discuss the fetal placental unit. And in terms of locations, by now you probably will remember that the adrenal cortex in the 3 zones, the zone of fasciculata, glomerulus, and reticularis, the ovaries and testes, and the placenta are all capable of synthesizing steroid hormones. And We went through a very tedious process of making all these different hormones from cholesterol, with all those hydroxylations and dehydrogenations and aromatizations and reductases. And hopefully now you have a better understanding of how to make these hormones, and you're on your way to understanding the clinical relevance and disease states that'll come later. And that concludes this video on steroidogenesis.